In 2026, inside a genetics lab at Oxford, researchers finished sequencing the DNA of 102 living men, all tied to a single remote stretch of the Greek coastline. What they found was a Y chromosome pattern that shows up almost nowhere else on the planet. The men in that sample have always believed something specific about themselves, that they descend directly from the Spartans. The lab could prove one half of that story. Centuries of near-total isolation, a genetic population sealed off from the rest of Greece. A signature so concentrated it stands out even against the broader European genetic map. But the other half of the story, the actual Spartan bloodline, remains unconfirmed, because no scientist has ever recovered verified ancient Spartan DNA to compare it against. There is nothing to check the legend against yet, and that gap is not an isolated curiosity. It sits inside a much larger decade of ancient DNA research that has quietly dismantled the idea that the ancient Greeks were ever one bloodline at all. Minoans, Mycenaeans, foreign mercenaries buried in mass graves, colonists who carried their genes across the Mediterranean into Sicily and southern Italy. None of them line up with the single unbroken lineage most people were taught in school. This is a tour through all of it. Crete, where Europe's first writing system was born. The Peloponnese, where that isolated population is still being studied today. Sicily, where a battlefield gave up secrets no historian ever wrote down. And a mountain range in Italy where villagers still speak a language descended from ancient Greek. Before any of that, there's one question worth sitting with, because it's the one this entire film keeps circling back to. What does it actually mean, biologically, to have Greek DNA? If genetics and ancient history in the same breath is something worth your time, subscribing costs nothing, and this channel keeps digging up exactly this kind of case. Because the myth this next segment takes apart is older, bigger, and far more comfortable than the isolated case in Mani. There's a version of Greek identity that gets repeated so often it barely gets questioned anymore. Modern Greeks as the unbroken genetic continuation of Pericles' Athens and Leonidas' Sparta, essentially unchanged for 2,500 years. Tourism brochures lean into it. Textbooks lean into it. Sparta gets leaned on hardest of all, precisely because so little physical material survives from classical Sparta to check the claim against in the first place. The actual ancient DNA record tells a messier story. Instead of one frozen population sitting untouched since the Bronze Age, it shows distinct ancestral layers stacked on top of each other over thousands of years. Early Neolithic farmers from Anatolia, migrants carrying ancestry linked to the Caucasus, steppe pastoralists arriving later, and centuries of additional Mediterranean-wide mixing on top of all of it. Even the two most iconic Bronze Age Greek cultures don't hold up the way 20th century archaeology assumed. For decades, many researchers treated Minoans and Mycenaeans as unrelated populations entirely, one framed as invading outsiders, the other as native islanders, two separate peoples who happened to share a region. Ancient DNA overturned that assumption rather than confirming it. Modern Greeks do sit closer, genetically, to the Mycenaeans than to the Minoans, according to the 2017 research that first tested this directly. But even that closer relationship comes with a documented dilution of the original Neolithic farmer ancestry, spread out gradually over more than 3,000 years. That's the tension running underneath everything in this film. An origin story people are certain about, sitting next to an origin story a laboratory can actually put to the test. The paper responsible for that first real test wasn't chasing a myth to bust. It wasn't built around Sparta, or a national identity question, or a documentary hook. It started with something much narrower and much more technical. A research team trying to figure out, as precisely as 90-year-old ancient bones would allow, exactly who had been buried on Crete thousands of years earlier. In 2017, a research team led by Iosif Lazaridis, working alongside David Reich, Johannes Krause, and George Stamatoyanopoulos, published a paper in the journal Nature titled Genetic Origins of the Minoans and Mycenaeans. Getting there required recovering genome-wide data from 19 ancient individuals, a genuinely difficult technical achievement, since the Mediterranean's warm, humid conditions are among the worst environments in the world for preserving ancient DNA over long stretches of time. The sample set itself was carefully built. Minoans recovered from Crete, Mycenaeans from the Greek mainland, a Neolithic individual from mainland Greece for comparison, and Bronze Age individuals from southwestern Anatolia to anchor the eastern side of the picture. The headline result reshaped how these two civilizations get discussed. Minoans and Mycenaeans turned out to be genetically very close to one another. both drawing at least 3/4 of their ancestry from early Neolithic farmers who had settled western Anatolia and the wider Aegean region long before either civilization existed. Both groups also carried a smaller, additional ancestry component tied to the Caucasus and Iran, evidence of an early eastward connected migration that had already reached the Aegean before Minoan and Mycenaean populations even split apart from each other. The one clear point of separation between them showed up specifically in the Mycenaeans, an extra 4 to 16% of their ancestry traced to northern populations, either Eastern Europe or Siberia, a component the Minoan genome simply did not carry. Krauss later described the motivation behind the project in direct terms. The team wanted to know whether Minoans and Mycenaeans were actually genetically distinct populations or not, who their respective ancestors had been, and how modern Greeks today relate back to either group. That last question would take years of additional research to answer fully. The first place to look for that answer starts on Crete itself, where the Minoans built Europe's first writing system, a script still sitting on clay tablets today that nobody has ever fully managed to read. The Minoan civilization took shape on Crete beginning around 2600 BCE, eventually producing Europe's first literate society through Linear A, a script still undeciphered to this day. When 19th century excavators first uncovered Minoan sites, many assumed the civilization's sophistication meant its people had to be foreign arrivals, too advanced, in the thinking of the time, to have developed locally out of an existing Aegean population. The 2017 genome data told a different story. Minoans trace back to the same Neolithic farming population that had already been established in the region for thousands of years, not a late wave of outside settlers arriving with civilization already built. Minoan genomes carry that same Caucasus and Iran-linked ancestry component found across the region, meaning some contact with populations to the east had already happened well before Minoan civilization reached its palatial peak on Crete. What Minoan genomes conspicuously lack is the northern, steppe-linked ancestry that shows up later in Mycenaean genomes, a clean genetic boundary separating the two cultures despite centuries of trade, artistic exchange, and shared regional history. Minoan writing, religious practice, and artistic style all point toward sustained contact with the Near East and Egypt. Genetically, though, the population itself stayed rooted in that same Aegean Anatolian farmer base the whole time, largely unmoved by the cultural currents flowing through it. The Linear A tablets themselves, some dated as far back as the 15th century BCE and housed today in the Heraclean Archaeological Museum, remain linguistically opaque even as the genetic picture behind the people who wrote them keeps getting sharper. 90 miles to the north, across open water on the Greek mainland, a population that looked broadly similar on the surface was about to reveal one specific, measurable genetic difference, the single clearest line separating two civilizations that later generations of archaeologists had once assumed were entirely unconnected to each other. The Mycenaean civilization developed on the Greek mainland starting around 1700 BCE, eventually extending its reach over Crete and the surrounding islands. Mycenaeans used Linear B, a genuine early form of the Greek language, distinct from Crete's still undeciphered Linear A, giving historians a direct textual thread that connects forward to the Greek spoken today. Genetically, Mycenaeans share the same core 3/4 Neolithic Anatolian and Aegean farmer ancestry found in the Minoans. The additional 4 to 16% of northern ancestry present in Mycenaean genomes stands as the single clearest marker separating the two Bronze Age cultures at the genetic level. For over a century, many archaeologists treated Minoans and Mycenaeans as unrelated, sometimes even framing them as rival populations locked in competition rather than close relatives. The genetic data overturned that framing entirely. Not two separate peoples locked in rivalry, but close relatives separated by one traceable, quantifiable point of difference. Modern Greeks, according to that same 2017 study, sit measurably closer to the Mycenaeans than to the Minoans, a specific and testable line of continuity running from Mycenaean-era mainland Greece forward toward the present-day population. Mycenaean archaeological sites also gave the modern world its only pre-classical window into Greek history outside of Homer's epics, since the Iliad and Odyssey described a world that archaeology only began to confirm as historically real in the late 1800s, when Mycenaean ruins were first properly excavated. That northern ancestry component, sitting at 4 to 16%, left one obvious problem the 2017 paper couldn't fully resolve on its own. It confirmed the presence of that outside ancestry, and it confirmed roughly how much of it there was. It did not pin down exactly where that ancestry originated, or precisely when it arrived in the Aegean, questions that would require an entirely separate body of research, years later, to finally answer. That question about origin and timing finally got answered years later, in 2021, when Florian Clemente and his colleagues published a new study in the journal Cell. Their team sequenced 6 new whole genomes, spanning the early to middle Bronze Age, along with 11 mitochondrial genomes, drawn from Cycladic, Minoan, and Helladic Mycenaean sites across the Aegean. What those genomes revealed first was continuity, not disruption. Early Bronze Age genomes from across the entire Aegean turned out to be remarkably homogeneous, derived mainly from the same local Neolithic Aegean ancestry that had already been established in the region for thousands of years. That result directly contradicted an older, once popular theory in archaeology: the idea that the shift from Neolithic culture into Bronze Age culture in Greece had been driven by a massive population turnover, one people wholesale replacing another. The genomes said otherwise. Continuity, not replacement, was the pattern written into the DNA. The northern, steppe-related ancestry that Mycenaeans would later carry only shows up substantially in Middle Bronze Age individuals from northern Greece specifically, and when it does appear, it isn't a faint trace. It sits close to 50% of their total ancestry. Clemente's team dates this steppe-related influx to around 2500 BCE, meaning it arrived as a distinct, later, geographically specific event, rather than a single uniform wave sweeping through every Bronze Age Aegean population at once. That timing also lines up with independent archaeological evidence of contact between southeastern Europe and the Pontic-Caspian steppe region during roughly the same period. Genetics and material culture pointing toward the same conclusion from 2 completely different directions. Put together with the earlier findings on Minoans and Mycenaeans, the picture that emerges is layered rather than simple. Local continuity first, across the whole region, followed by a targeted northern migration arriving unevenly, touching some populations far more than others, rather than moving through the Aegean as one single sweeping event. 50% steppe ancestry in the north. Homogeneous Neolithic ancestry everywhere else in the early Bronze Age. Same region, same rough era. 2 entirely different genetic stories. There was a third Bronze Age Aegean civilization sitting inside Clemente's dataset alongside the Minoans and Mycenaeans, one far less famous outside of art history circles, the Cycladic culture. Genetically, it belongs to the same early Bronze Age homogeneous population as its more celebrated neighbors. Cycladic figurines and marble artwork remain instantly recognizable today, staples of museum collections and modern design references alike. Yet the people who carved them shared the same Neolithic Aegean and Anatolian ancestral base as the populations living on Crete and the Greek mainland at the same time. The 2021 Cell paper states this directly, describing Cycladic, Minoan, and Helladic populations as genetically homogeneous across the Aegean and Western Anatolia at the very beginning of the Bronze Age, despite the fact that each of these 3 cultures went on to produce strikingly different material traditions. That's a specific and testable claim sitting underneath the archaeology. Shared genetic ancestry did not translate into shared artistic style, religious practice, or political organization. 3 civilizations, diverging sharply in how they built, worshipped, and governed, all starting from the exact same genetic pool. The paper's authors also note something about the state of the field before their own work. A long-standing shortage of genomic data, specifically from the Neolithic to Bronze Age transition period, had left this particular question open for years, unresolved simply because nobody had recovered and sequenced enough material from that narrow window of time. Their new sequencing filled that gap directly. Same starting population, 3 different civilizations. The next chapter of this story unfolds 1,000 miles away, on a battlefield where nobody working in Greek history ever expected to need Siberian DNA to explain what happened. By 750 BCE, Greek city-states had begun founding colonies across Sicily and southern Italy, a region the Greeks themselves referred to as Magna Graecia, and the wave of settlement came from multiple, named points of origin. Euboeans founded Pithecusae, and Cumae around 750 BCE. Spartans settled at Tarentum. Achaeans founded Metapontum, Sybaris, and Croton. Locrians established Locri Epizephyrii. Chalcidians founded Rhegium. Among these new settlements sat Himera on Sicily's north coast, founded by Ionians and Dorians around 648 BCE, and it would go on to become one of the westernmost Greek footholds anywhere in the Mediterranean. Himera's location put it directly in the path of Carthaginian power, and the city hosted 2 decisive battles against Carthage. A Greek victory in 480 BCE, followed by a catastrophic loss in 409 BCE that ended Himera's existence as an independent city-state entirely. Ancient historians wrote about both battles in detail. Herodotus, 29 years after that celebrated 480 BCE victory, Himera faced Carthage again in 409 BCE, and this time, according to the ancient sources, the city stood entirely without allied reinforcements. Genetic data from the soldiers who died defending Himera in this second battle tells a completely different story from the first. Their ancestral origins were exclusively local, Sicilian and Greek, with none of the Northern European, steppe, or Caucasus signatures that had turned up in the 480 BCE dead. Himera lost that second battle. The city was destroyed and never again functioned as an independent city-state, a documented historical endpoint that the genetic evidence lines up with precisely. The absence of diversity in this second set of remains is itself the evidence worth pausing on. No reinforcements arrived, no foreign mercenaries fought alongside the defenders, no allied city sent support. Just local defenders, standing alone, facing an enemy that had beaten them once before and would not be turned back a second time. Taken together, these two battles give historians something genuinely rare in the study of ancient warfare. Two dated conflicts from the exact same city, separated by less than 3 decades, one genetically diverse and victorious, the other genetically homogeneous and catastrophic. It functions almost like a natural experiment, the kind ancient warfare essentially never preserves this clearly. The takeaway isn't that mercenaries guaranteed victory on their own, and the genetic record doesn't claim that either. The real significance is narrower and more concrete. The presence or absence of outside manpower in an ancient army is now something a laboratory can measure directly, pulled straight out of a body count that the written historical record stayed completely silent about. Himera's story as an independent polis ended in 409 BCE, sealed by defeat and destruction. But along that same Sicilian and southern Italian coastline, other Greek colonies founded during the same centuries of expansion were still very much active, their own populations leaving a genetic signature that researchers would eventually trace back with just as much precision. Sergio Tofanelli and his colleagues published a study titled The Greeks in the West in the European Journal of Human Genetics in 2016, taking on a question that had lingered, largely unresolved, for far longer than the Himera battles. How thoroughly did Greek settlers actually replace or mix with the existing population of Sicily and southern Italy during the colonization era? The competing theories the paper lays out at the start span a wide range, from small groups of Greek male colonists lightly mixing into indigenous communities, all the way to substantial migration, amounting to genuine demographic replacement across the region. To test between those possibilities, the team worked with a sample size of more than 800 people, split between reference populations drawn from Euboea and Corinth, the historically attested source regions for the first wave of colonists, and people whose families are native to Sicily and southern Italy today. Earlier studies in this space had relied on single Y chromosome lineages as colonization markers, an approach Tofanelli's team deliberately avoided, since modern genomes simply don't preserve markers that specific with any reliability across more than 2,500 years of mixing and mutation. Instead, using both Y chromosome and mitochondrial DNA, alongside demographic modeling of mutation rates over time, the team recovered a clear signature of Greek paternal contribution to Sicily, dated specifically to the Archaic period, running from the 8th through the 5th centuries BCE. That dating lines up precisely with the historically recorded founding waves covered earlier: Euboean, Spartan, Achaean, Locrian, and Chalcidian colonies, all established within that same several-century window. That genetic signal comes through strongest in the coastal cities, the places where those original colonies were physically founded. Head inland instead, up into Calabria's mountains, and a much smaller, much stranger community carries a related but distinct version of this same story. Stefania Sarno and her colleagues published a genome-wide study of Calabrian Greeks in Scientific Reports in 2021, focused specifically on communities isolated deep in the Aspromonte Mountains of southern Italy. These are the communities that still speak Greco today, a language variety directly tied to the ancient Greek presence in the region, a linguistic fossil that has managed to outlive the political history of Magna Graecia itself by well over 2,000 years. Sarno's study used 720,000 genome-wide markers, a substantially higher resolution follow-up to an earlier characterization of the same population that had relied on roughly 150,000 markers. The results show measurable genetic drift and long-term reproductive isolation specific to these Aspromonte communities, setting them apart from the surrounding, non-Greek-speaking Calabrian population living in the same general area. The isolation itself is the core finding here. Geography, not just written history, is what allowed a linguistic and genetic signature this old to survive intact into living communities in the present day. Broader Mediterranean-wide studies covering Sicily and southern Italy add further texture to this picture, identifying post-Neolithic Levantine and Caucasus-related ancestry threaded through the wider region, consistent with the same maritime Bronze Age contact patterns that had already shaped the Aegean itself centuries earlier. Calabria kept a language alive through isolation, preserved in mountain villages far from the coastal cities where Greek colonization first took hold. Back in Greece itself, a single peninsula kept something else alive through that same kind of isolation. And it took until 2026 for a genetics team to actually prove it. The Mani Peninsula juts off the southern edge of the Peloponnese, one of 3 narrow fingers of land extending from mainland Greece into the sea, rugged and mountainous. Historically part of ancient Laconia, the territory Sparta dominated from the 7th century BCE onward. Under Roman rule, Mani retained a degree of autonomy inside the League of the Free Laconians, active from 195 BCE through 297 CE. After that period, the historical record goes almost completely silent. Aside from a successful defense against a Vandal invasion in the 5th century CE, essentially nothing substantial was recorded about the region's population for roughly the next 400 years. Byzantine Emperor Constantine VII, Porphyrogenitus, later described the people of Mani in unusually striking terms, language that suggests historians of his own era already suspected this region had followed its own separate path, distinct from the rest of Byzantine Greece. The Deep Maniots today are known for a distinctive local dialect, a strict patriarchal clan structure, and stone tower houses that have drawn travelers and writers for generations, including Jules Verne and Patrick Leigh Fermor. A long-standing local oral tradition claims something more specific still, that Deep Maniots descend directly from the ancient Spartans, and that the region continued worshipping the Olympian gods long after the rest of Greece had converted to Christianity, by some accounts as late as the 9th century CE. That's the legend, passed down through generations, tied to stone towers and an isolated dialect and centuries of near-total historical silence. In 2026, a genetics team finally set out to test how much of it actually holds up. Leonidas Romanos Davronoglu led an international research team, drawing on the Oxford University Museum of Natural History, Tel Aviv University, the National and Kapodistrian University of Athens, and the genealogy company Family Tree DNA, publishing their results in the journal Communications Biology in February 2026. The team sequenced Y-DNA and mitochondrial DNA from 102 deep maniots using next-generation sequencing methods. Roughly 80% of the men sampled carry the West Asian Y haplogroup JM172, also known as J2A, an exceptionally high concentration for any single population anywhere in Europe. Within that here's the specific gap sitting underneath everything else the 2026 study found. The Deep Maniot data proves a genetic island stretching back roughly 1,400 to 1,600 years. It does not, on its own, prove descent from the Spartans specifically. Study co-author Athanasios Kofinakos addresses this directly, noting that no confirmed ancient Spartan DNA currently exists anywhere in the world. There is no genetic reference sample from classical Sparta itself against which the legend could actually be tested. The Spartan descent claim remains a specific, testable hypothesis waiting on a sample that simply doesn't exist yet. It is neither rejected nor confirmed by this research, just left open. Independent commentary points to a second limitation running alongside the first. Archaeologist Chelsea Gardner of Acadia University notes that 102 sampled individuals is a relatively small dataset, and that a larger sample could still reveal connections to other populations the current data isn't sensitive enough to detect. Gardner's own framing is worth stating precisely. The study is genuinely interesting, but on its own it does not prove any cultural or historical reality. A caution aimed squarely at the gap between measurable genetic isolation and the specific historical narrative people choose to attach to it. What the study does establish, independent of the Sparta question entirely, is a documented Bronze Age through Roman genetic continuity that predates the 6th century CE migrations that reshaped the rest of Balkan demography. Deep Mani was largely insulated from precisely the population movements that changed nearly everyone else living around it. Every case covered in this film runs into some version of that same gap. A real, measurable genetic pattern sitting next to a story about that pattern that genetics alone can't fully settle. Step back across every population covered so far, and one mechanism keeps repeating itself in the data. Genetic continuity survives specifically in places where geography or social structure limits who breeds with whom over long stretches of time. Deep Mani's mountainous terrain and strict patriarchal clan system represent the clearest version of this mechanism at work, limiting outside contact for men specifically, while allowing more mixing for women, sustained over roughly 1,400 years. Calabria's Greco communities show the identical pattern operating at a smaller scale, mountain isolation in Aspromonte, preserving both a language and a distinguishable genetic signature against a numerically much larger surrounding population. Himera demonstrates the exact inverse mechanism, playing out in real time within a single historical moment. As a cosmopolitan trading and military hub, the city pulled in ancestry ranging from the Caucasus to the Baltic within one battle, a direct reminder that war and colonization function as population mixing events, not isolating ones. The Bronze Age Aegean data shows both mechanisms operating within the same region, at different moments in time. Homogeneous local continuity defined the earliest Bronze Age, followed by a targeted, geographically uneven steppe migration, arriving specifically in the north, around 2500 BCE. Founder effects. The genetic bottlenecks that follow a population crashing down to a small number of surviving individuals show up with specific estimated dates in the Deep Maniate data, placed at 380 to 670 CE, precisely because sustained isolation afterward locked that reduced diversity in place, instead of letting it gradually dilute back out over the following centuries. Which raises the question this whole film has been circling since the opening scene. Zoom back out to the national picture, and Lazaridis' 2017 study places modern Greeks overall closest to the Mycenaeans among every ancient population sampled, alongside a documented dilution of the original Neolithic farmer ancestry accumulated over more than 3,000 years. That dilution isn't some unexplained erasure. It is the accumulated record of steppe migration, Roman-era mobility, Byzantine-era population movement, and centuries of Mediterranean-wide contact, all layering steadily on top of the original Bronze Age base. Regional variation within modern Greece is itself now a measurable, mapped phenomenon, with Deep Mani sitting at one isolated extreme and other regions showing far more blended ancestry from exactly the population movements Deep Mani was specifically shielded from. The same pattern echoes well outside Greece's current borders. The Greco of Calabria and the descendants of Magna Graecia's colonists in Sicily carry a Greek genetic signature dating specifically to the 8th through 5th centuries BCE, sitting today inside an Italian national identity rather than a Greek one. None of this erases Greek identity or cultural continuity in any way. Greek language, religion, and tradition demonstrably persisted straight through every one of these population shifts. It simply means the genetic story sitting underneath that identity is a layered composite built up over thousands of years, rather than a single unbroken thread running back to one founding population. The myth said one bloodline. The data says something closer to a very long relay race, and the baton got passed more times, in more directions, than anyone had assumed. No confirmed ancient Spartan genome exists yet anywhere in the world, meaning the single most requested comparison sample in this entire body of research remains unrecovered. The Deep Maniate sample size, at 102 people, is large enough to establish a genuine pattern, but small enough that outside researchers are already calling for an expanded dataset before treating the isolation finding as fully settled science. The 2021 Cell paper's own 6 new Bronze Age genomes represented a major addition to the field at the time, yet they still amount to only a small fraction of the total burials known from Cycladic, Minoan, and Helladic sites scattered across the Aegean, meaning plenty of populations from this same era remain completely unsequenced. The Himera mass graves gave researchers a rare, precisely dated two-battle comparison, a research situation that's almost impossible to replicate for most ancient conflicts, since it depends entirely on unusually well-preserved and unusually well-documented mass burials surviving intact for over 2,000 years. Every single population covered across this film was reconstructed from a specific, finite sample, ranging from dozens to low hundreds of individuals, standing in for entire populations that, at the time these people were alive, numbered in the thousands or considerably more. Which is exactly why the next study on any one of these regions could change the picture again. Back in that Oxford lab from the opening scene, 102 Deep Maniate samples, an 80% concentration of a single Y haplogroup, and a legend about Sparta that the data supports in shape but still can't confirm by name. Across this film, Minoans and Mycenaeans shared a Neolithic base with one traceable northern addition. Steppe ancestry arrived late, and unevenly, touching some populations far more than others. Himera's soldiers carried the Mediterranean's entire mobility network into a single battlefield, then vanished from the record entirely by the Second Battle. Magna Graecia's colonists left a Y DNA signature still measurable in Sicily and Calabria today. Deep Mani's isolation locked in a genetic profile the rest of Greece lost centuries ago. The through line connecting all of it is direct. Every one of these populations believes something specific about where they came from, and in every single case, the genetic reality turned out more layered, more mixed, and more geographically specific than the story that got told about it. So the open question worth sitting with is this: if the most isolated, most mythologized population in this entire story still can't get full genetic confirmation of its own legend, what does that say about the confidence everyone else places in theirs? The door stays open rather than closing here. More Bronze Age genomes, a hoped-for ancient Spartan sample, and ongoing work in Calabria and the Peloponnese all mean this specific question is still being actively answered, not finished. The labs are still working. And the next name on this long list of settled ancient origins could belong to any one of the populations just covered here.